That is, the chick chorioallantoic membrane was exposed by cutting a window through the eggshell, and experiments were carried out in ovo, resulting in significant limitations in the accessibility of the cam and in the possibilities for observation and photodocumentation of effects. Ex ovo cultures of chick embryos significantly facilitate the accessibility of the cam and the chick embryo, please see the beating heart, enabling easy in vivo documentation of effects and experimental manipulation of the embryo. In our video article, we are going to provide a step-by-step demonstration of the successful application of ex ovo culture for a large number of scientific. The elbows should not rest on the bench top. Autoclaved filter papers are used as supporting material for liquid substances applied to the cam, as they seem to cause the least irritation of the cam. The application site should be halfway between the embryo and the border of the cam, as otherwise the embryo hinders the microscopic observation of effects in transmitted light. The liquid, here angiotropin, should be applied immediately after the filter has been placed onto the cam. To avoid drying, either additional doses or buffer should be reapplied every day. When the cam is fully developed, up to 6 different samples may be applied and their effects can be compared on a single cam. Rings cut from a 1 milliliter pipette tip should be cut as thin as possible to minimize weight and to avoid indentation of the cam. The ring is then applied to the cam and the cells are pipetted into the ring. The microsyringe is filled with the prepared cell suspension. Carefully but quickly penetrate the cam and the eye layers with the syringe needle and continuously inject the cell suspension. The needle should remain some seconds in the eye to avoid a loss of the injected cell suspension by leakage. Chick ex ovo cultures of eggs incubated for 3 days are used as limb bud donors. The embryo is dissected from the connected yolk vessels by cutting a circle. The embryo is transferred to a petri dish filled with cold, sterile PBS by use of a small drained spoon and washed by agitation. The embryo is then transferred to a fresh petri dish filled with sterile PBS and freed from the surrounding membranes, carefully tearing them apart with a fine forceps. The limb buds are detached by forceps as close to the body as possible, grasped and transferred to the cam of an 8 to 10 day old host chicken. At the desired application site, the cam is selectively traumatized by gentle scraping with one chunk of the forceps. Then the graft is grasped and layered onto the cam with the other chunk of the forceps. The sacrificed female mouse is fixed on a board. The abdominal wall is moistened with 70% ethanol, cut along the midline, and the skin flaps are fixed laterally by pins. The uteruses are removed from the abdomen, detached, and transferred to a beaker with cold PBS. The embryonic membranes are removed carefully by the use of forceps. For demonstration purposes, a 16-day-old mouse embryo is shown here for better visibility of the limb buds. Optimal grafting results are only obtained, however, if limb buds from 10 to 13-day-old embryos are used. The limb buds are cut off by the use of fine forceps as close to the body as possible. At the desired application site, the cam is selectively traumatized by gentle scraping with forceps. Then, the limb buds are grasped and transferred to the cam of an 8 to 10-day-old host chicken. The graft is placed once or twice on a side of the cam where no final grafting is intended to remove excess PBS. The grafts are ideally placed on the cam near a Y bifurcation of a blood vessel. Tumor biopsies are cut into 1 to 2 cubic millimeter pieces by sterile scalpels. Taking material from the surface of the biopsy increases the risk of contamination with muscle or connective tissue. At the desired application site, the cam is selectively traumatized by gentle scraping. A piece from the core of the biopsy sample is grafted onto the cam of an 8 to 10-day-old host chicken. The grafts are ideally placed on the cam near a Y bifurcation of a blood vessel. The graft is then transferred to the cam with minimal PBS attached. Light pressure might be applied to maneuver the graft into a resulting indentation of the cam. The host chick embryo is killed by decapitation. The cam with the attached graft is removed by a circular cut and transferred to a petri dish filled with PBS. Excessive cam is cut from the graft except for the former attachment site. Again, at the desired application site, the cam of a new host chick embryo is selectively traumatized by gentle scraping, and the graft is transferred to the cam of the second host with minimal PBS attached.